Okay. I'm going to speak in English because we have an audience today. But you can, of course, stop me and ask me questions in Spanish or in Portuguese whenever you want. But later, maybe we can edit the video to make it available for a broader audience. So I'll talk to speak in English. Do you hear me well? Okay. I'll make a brief. I'll do a brief introduction about the complex mixtures package, which is a package to understand interactions in very complex molecular systems. So if you want to search for it, you can go directly to Google and type ComplexMixtures.jl. .jl because this is a package written in the Julia programming language, and all the packages in this language have this extension, .jl. And when searching for this package on GitHub, on Google, you'll get first the GitHub page, where you have the source code of the package and so on, and all the details, the technical details. And the second entry on Google, which we will take you to the docs, which is this link here or here. And the docs are the place where you should start reading to understand what the package does and how to use it. I'll start with a brief introduction of what this package does in general, and for that I'm going to draw something. So this is a package that computes a kind of distribution function which is not the most common distribution function, but I'll try to illustrate how these distributions work by doing a small drawing here. Can you see the board? So the general idea is the following. So you have any solute, and the solute might have a complex shape. So this is your solute. Typically, it's easy to speak about a protein, but that's not a necessity of the software at all. You can work with any kind of solids, but we have a lot of applications where these solids are protein, so I probably will say protein many times. And this protein has a complex shape, so it's far from being a spherical object. Around the solid, there is some other molecules, and for instance, water and maybe ions and something else here around this molecule. And we want to understand how these other molecules interact with the protein in both structural ways and thermodynamic ways. So usually what we do to understand this kind of interactions is to compute a distribution function. Distribution function is something that will tell us, for instance, how many molecules of water, because if we have water here, we'll compute something that is the density of water as a function of the distance of the protein. And this R is the distance relative to the protein. We normalize this distribution by an ideal distribution, which an ideal density. And this ideal density would be the density of water molecules that would be at each distance of the protein if there were no interactions of the water with the protein. I will speak a little bit about this later. So what do we get when we compute a distribution function like this? First, in the regions where the protein is explicitly occupies a volume, we don't have any water molecules. So there are no water molecules in a region very, very close to the protein up to a distance, which I will call here R0, which is the first distance where we start to see some water molecules. So this distribution function will be 0 because the density of water molecules inside the volume of the protein is 0, and the density of water molecules that would be there if there were no interactions between water and the protein is different from 0, because if there were no interactions between water and the protein, the water would be able to penetrate inside the protein and occupy this space. So here in this first region of small distances, we have a 0 density divided by a non-zero density. Then there is some short distance here where we start to find water molecules. And These water molecules at the shortest distances are probably forming hydrogen bonds. And the typical distance between the atom of the protein that's forming the hydrogen bond and the oxygen atom of the hydrogen bond of water or the hydrogen atom is of the order of about 1.8 angstroms. So at about 1.8 angstroms, we start to see the presence of water. And thus the density of water at these distances start to increase. And the density of water that would be there if there were no protein-water interactions is constant relative to the volume. So because of these interactions being favorable interactions, we see this ratio going to numbers greater than 1. So we start to see an accumulation of water at these distances, and we see a peak associated with the hydrogen bonding interactions, and this peak will be centered more or less at 1.8 angstroms, which is the typical distance of a hydrogen bond. Okay, then since this water molecule is forming a hydrogen bond here, so there is an intermediate region here where there is not another water molecule, and there will be a depletion region, and then there will be water molecules forming hydrogen bonds with the first water molecules, and then there will be another accumulation and so on. And at long distances, this distribution function converges to 1 because when one is far from the protein, there is no more correlation of the presence of the protein and the distribution of the solvent. So, for instance, here, far from the protein, from the solute, the density of water is identical to the density of water that would be there if there was no protein in the solution. So this ratio goes to 1. So this picture is obtained if we compute a specific type of distribution function, which is the minimum distance distribution function. So what I'm drawing here and what I'm illustrating is the distribution of the minimum distance between any atom of the protein, or if we want to say it less technically, the surface of the protein and the molecules of water. So here I have this 1.8 angstrom. Here is the distance between the atom of water which is closest to the protein surface, and here 1.8 angstroms at this point is also the distance that connects the closest atom of the protein with the closest atom of water. The closest atom of the protein with the atom of water that's closest to the protein. And this distance, the shortest distance, is the distance that we usually envisage when we think about molecular interactions. When we think, okay, the protein is forming a salt bridge with some other atom, with some ion in the solution, we have a picture in our minds, which is the charged amino acid residue forming a short distance interaction with that ion. And this occurs at a small distance. So computing these minimum distance distribution functions is what allows these distributions, which I call MDDFs, minimum distance distribution functions, to be interesting from the point of view of the interpretation of the molecular interactions, independently of the shape of the solid. So even if it doesn't matter the fact that this solid, this protein, has a very complex shape. We see here that the protein has a very complex shape. It doesn't matter because we are computing the minimum distance between the other and the protein, and the first peak will appear at the distance where hydrogen bonds are occurring, and other peaks will appear where other interesting interactions are occurring at the surface or nearby the surface of the solid we are investigating. So the whole package is built around this idea of computing minimum distance distributions. The minimum distance and trying to understand the structure of the solution by the distributions of the minimum distance between the atoms of the solutes and the solvents. And then, given that, we have an additional feature of these minimum distance distributions. For example, we have here a hydroxyl atom of the protein. And then we have a water molecule here. And okay, here, I draw it in the wrong position. Let's draw it forming a hydrogen bond. I have another water molecule here. And I can, when I compute the minimum distance distribution, at this point, I have noticed that the atom of the protein that is satisfying the minimum distance to these water molecules is this oxygen atom. So this oxygen atom is the one involved in the minimum distance relative to this particular water molecule at this point in my simulation. Then I can annotate that. So when in the process of computing this minimum distance distribution function, my package will annotate who is this atom that is satisfying this minimum distance at this point. So who is this? This is the oxygen OH from the serine 43 of the protein. And this information will be stored when I compute this distribution. And we'll see why this is interesting and how we will obtain very rich information about the molecular interactions by storing this information. At the same time, at this point, I have annotated that this H2 atom of the water molecule is the atom of the water molecule satisfying this minimum distance. Then what will happen is that afterwards, when I end the process of computing all the distributions, I will be able to decompose this distribution function, which is the global distribution function that I have drawn here. I will be able to decompose the distribution function in the contributions of the atoms that were the ones satisfying these minimum distances at each point of my simulation. So I will be able to get this distribution function and see which is the contribution, for instance, of this serine 43 atom, of oxygen atom of the serine 43, and then with this information know if the water molecules were interacting with this particular atom or this particular residue or this particular any kind of decomposition of the structure in any subgroup of the structures of the solute or the solvent. So we will see this in the examples that I will be showing now. So this is the general idea. Everything is centered around computation of minimum distances, annotating which minimum distances are necessary for these interactions, for understanding these interactions. And now we are going to start one example, and then the example will illustrate what I'm saying in practical terms. Is everything more or less clear for now? So now, now that I explained more or less, it's probably easier for you to understand the introduction of we should focus now on this screen. Let me see. Share the screen. I don't know what happened here. Okay. Okay. Now you are seeing. Okay. So now that you have this introduction, it will more or less explain the same thing that I have mentioned now, and explaining these features of the distribution function that we are computing here. And now we will going to see an examples how to apply. This is one important thing about is the help section. You can ask, of course, things about this package, sending emails to me. We have a Julia chat. If you want, you can join this chat if you want to ask questions without sending an email. Opening issues in the package, that may be very common, and you can also ask questions in the Julia Discourse forum, particularly if the questions are not very specifically related to the package, but with the use of the package. I'm very active in this forum, so if you ask something about my package there, particularly if you mark me, I will be answering probably quickly. Okay, so what I'm going to show you here in practice, I will follow the examples of the page. So here in the page, in the documentation of the package, I have a series of examples, and I will follow these examples but running them locally here in my machine. But in general terms, the codes that I would be executing here in my machine are simply the codes that are available in the scripts of the example. So you don't need necessarily to be anxious about annotating the commands and so on, because everything is exactly what's explained in these examples. And later, if we have time, I will show you a little bit how to work with the Zika virus, which has some peculiarities and is not one of the examples of the page, of course. So to present, I will start with this protein in water glycerol system, which is the most typical kind of system that we studied with this package. And what we have in this system is the protein. I don't remember which protein, and this is solvated by a mixture of water and glycerol. So what we have is what we need to perform all the analysis of the package is a PDB file of the system, which here it's called system.pdb, and a trajectory file. Okay? Here I have a trajectory 1 GB file, which is a reasonably large enough to actually perform some interesting analysis. And in this case, the file is a DCD file generated by an AMD, but the package accepts Romax, Amber, and so on trajectory files, and then any kind of trajectory file, so that's fine. So now I'm going to share. I'm going to share my screen here. Now, what you are seeing is my Visual Studio Code page. Okay, and what we have here is, okay, so how do we get to this point? Here I'm using Julia. Here I'm using the Visual Studio Code editor because it's convenient, but this script can be run and can be edited on any text editor and can be run on the command line. This is actually the way that the page explains running the script is from the command line. It's reasonable, but it's better when one's learning to use the package to actually run each command one by one to understand what the commands are doing, so we can follow more closely what's happening. So one thing about Julia in general, here I started a Julia terminal, which is more or less the same thing as a Python terminal, but the Julia terminal and the Julia REPL includes package manager. So if you type the close brace sign in your Julia package, in your Julia REPL, it will go to this package prompt, which is the package manager of Julia. So here is where you can add packages. For instance, I can add the complex mixtures package here. Of course, here it will be already installed, so it won't do anything. But the package can be installed by typing on the Julia REPL close brace, which will automatically bring you to the package manager, and that's the package. Alternatively, you can use a command like this one, which is the way to explain it in the manual. But this is more or less usually in Julia we work in the REPL because the REPL is very, very practical. Additionally, here the first two commands that I posted here is to activate the current directory. So here it's already activated, but if I type it here, activate dot, I'm activating a project in the current directory, which means that I will be installing packages. This package will be installed in this project. It's more or less like in environment, independent environment in Python or in other languages. So once you install the package in this directory, you have a reproducible state for your script and so on, and you won't be needing to reinstall the package again. And if you save these two files, the project and manifest files that are available here in the directory, here, project and manifest file, you can, if you save them, you can be sure that at any time in the future, you'll be able to reproduce the scripts and rerun the scripts that you are producing here with this exact same version of all the packages you have used once you have installed. So anyway, I consider that we have already installed this package. This is, of course, the ComplexMixtures package, ComplexMixtures package. The PDB tools package is also a package that we developed to manipulate PDB files. This is the plots, the most common package for producing figures in Julia, and this is the latex strings package to write labels. The plots use latex. But actually, in the examples, I'm not going to explicitly use them. So anyway, what do we have? What do you need to? So here we are going to using ComplexMixture. Using is the command for use package in Julia. Using ComplexMixtures, PDB tools, and plots. These are the ones that we are going to be using here. And then we are going to read the PDB file, which is available. This is a PDB file of the complete system, anyone. And when you read this PDB file, you see that we have a vector of atoms with, in this case, 130,000 atoms. And okay, these are the atoms. We have protein, and then we have the glycerol residues afterwards in this. So then we are going to split these atoms into the protein and first the glycerol. So we are using a selection tool of the PDB tools package here. So we are going to select the protein atoms of the PDB file. So now we have a vector of 3,800 atoms, which is the protein in the structure, and the glycerol. I'm just following the exact comments of this script, explaining a little bit one by one what they are doing. So, and we select the residue names, the residues that are named with glyc, which are the glycerol residues of this structure. Given the selection of the protein atoms and the glycerol atoms, we are going to define what ComplexMixtures needs to understand what's the solute and what's the solvent of your system. So you have a solute with an atom selection protein, and we are saying, okay, this is the solute, and this solute has only one molecule. So this is the command that informs the ComplexMixtures package that the protein is a single molecule. This is, we'll see in further examples that you can have solids formed by more than one molecule and still use an MOS-equal and MOS-equal one, depending on what we want to compute. But here we have, okay, these atoms belong to a single molecule, and similarly, we are going to define the solvent as an atom selection of the glycerol residues. But here we have, if I remember correctly, 14 atoms per glycerol. So if you read this, okay, we have 60,000 atoms that belong to this number of glycerol molecules, and this we have to inform the ComplexMixtures package that our solvent is formed by a multitude of molecules of the same size of 14 atoms. Then we have a trajectory file, which is the NAMD generated trajectory file that we have here. DCD. This is the trajectory file. And now we go directly to the main function of the package, which will run the analysis that I'm talking about. So, and I will explain a little bit what this does range. Okay, so essentially this will compute the distribution functions and all the data required for more specific analysis of these distribution functions on this trajectory, where the solute is this molecule, the solvent is this molecule, and here we can set a very great variety of different options, but here we will explicitly set which region of the solution is the bulk range of the solution. This is an important parameter that I will explain now what it means. Okay, so I run this, okay, it will run. This will run in parallel. I'm running here in a relatively good machine. You see, we have 20 threads in this machine, and this is running. It will take about a minute to run on 667 frames. It will take about more or less about a minute, and while it computes, I will go back to my drawing board to explain a little bit what this bulk range is, because this is important if you want to compute thermodynamic properties of this solvation. So back to our, let me see what you see. Okay, back to our drawing board. We have our solute, and I defined a parameter that was called bulk range.
Mm-hmm.
Bulk range equal 10, 15. So what does that mean? Remember that I have to compute the distribution function, which is the density of water, in this case glycerol, at this distance, divided by the density of an ideal solution. How do we compute this ideal solution? This is the normalization that has to be done to actually compute meaningful and thermodynamically correct distribution functions. But this distribution function, this ideal distribution, is not trivial to calculate, because for several reasons. We have around this solute a region in which the presence of the protein, okay, here's the protein, the presence of the protein affects the density of the solvent in this region. This region nearby the protein, where the protein affects the structure of the solvent, is called, or it's generally called, the solute domain. So in this region, the protein affects the structure of the solution. Then we have, of course, our simulation box. And of course, outside the simulation box, we have the bulk solution, where the presence of the solution is far enough from the solute in such a way that the presence of the solute does not affect anymore the structure of the solution. Because the protein affects the structure of the solution, we can have, as we have shown, an accumulation. Here is an accumulation of the solute around the protein. Then if I have this accumulation, that means that the density of the solution outside of the protein domain is in principle unknown. Because even if I prepared my system in such a way to obtain a specific density of solvent molecules outside in the complete box, because there is accumulation of the solute of the solvent in this region, you might have a different density of solvent outside this region, depending on how much there is of solvent molecules inside nearby the protein. So if the protein interacts very strongly with your solvent, there will be accumulation of the solvent in the vicinity of the protein, and the bulk density will decrease. And the contrary is also true. If the protein repels your solvent, there will be more dense. The density inside the protein domain will be smaller than in the bulk, and then the density of the solvent in the bulk will increase relative to what you were expecting. So the first thing that we have to have in mind is that in order to obtain which would be the distribution of the solvent if there were no water-protein interactions or solute-solvent interactions, we have to estimate which is the density of solvent in the bulk solution in this simulation, which is not something that is given by the preparation of the system. It's something that results from the equilibration of the solvent around the solute. So we cannot know in advance the exact density of the bulk solution, of the exact concentration of the solvent before the simulation, because the arrangement of the solvent around the protein will change the distribution of the molecules in such a way that the final bulk density of the system is not the initial bulk density of the system which was prepared, but it's something that depends on the way the system interacts with the solid. So we have to estimate this bulk density to know how many water molecules or how many solvent molecules there would be in this region. This is the bulk solution. And we need to normalize the distribution function. We need to know how many solvent molecules there would be in this complete region if there were no solute-solvent interactions. But for this, we need to estimate the density of the bulk. So in order to estimate the density of the bulk, we could simply, we could, and this is one of the options of the package, compute, okay, how many solvent molecules are inside the solute domain? Let's call it N molecules of the solute domain. This is the solvent molecules, okay? This is the number of solvent molecules, glycerol or water molecules. This is the number of solvent molecules inside this region where the protein affects the solution. Of course, the number of molecules outside, the total number of molecules is the number of molecules outside the solid domain, I will write outside the solid domain, OSD, plus the number of molecules of the solid domain. This is rather trivial. So the total number of molecules that I have put in my simulation of the solvent is the number of solvent molecules that are inside the solid domain plus the number of molecules that are outside the protein domain. So this suggests that we can compute the number of molecules that is outside the solid domain simply by subtracting the total number of molecules from the total number of molecules, the number of molecules of the solid domain. Is that clear? So we could try to estimate the density of the bulk as the number of molecules outside the solid domain divided by the volume, total volume minus the volume of the solid domain. See, this would be the density of water molecule, of the density of the bulk solution. Except this is one possibility, except that this possibility is not ideal. Why it is not ideal? Because the number of molecules inside of the solid domain and the number of molecules outside of the solid domain are completely correlated. So you are not really measuring two different things if you are computing the density of molecules in the solid domain and the density of molecules outside the solid domain, because they are essentially the same. One is one minus the other relative to the total number of molecules. So there's a better way to estimate, which would be actually the uncorrelated bulk density of the solution, which is compute a new boundary here, define a new boundary here, and compute the number of molecules in the bulk range. There will be this new bulk range. I'll paint it another color here. So this is now with what we defined as the bulk range when we started the computation there. Which is, what's the bulk range? Is a region that is outside the solid domain, starts in the boundary of the solid domain. So we already believe that the presence of the protein does not affect the structure of the solution in this region. But it's an open system. What does it mean that it is an open system? The fact that it is an open system means that there are molecules that are being interchanged from the inside and outside of this domain all the time in the simulation. Molecules go in and out from this region, which means that from the point of view of this green region, this is a simulation. Is as if this was a simulation performed in the NPT and NPT ensemble, in the grand canonical ensemble. Which means that the number of this is an open system which has a bath of solvent molecules, which is feeding and interchanging molecules with this open subsystem of our system. And now the number of molecules that are inside this bulk range is not anymore directly correlated with the number of molecules of the solvent domain, but is the result of the equilibration of the solution in an open system in which can in fact interchange molecules with the solution in general. So the density of the bulk solution will be estimated by the number of the molecules of this bulk range, which is this one, divided by the volume of this bulk range. And the number of molecules of this bulk range and the volume of this bulk range are parameters that are determined essentially independently from the total number of molecules of your solution. So this is a little bit technical, but this is the best way from a thermodynamic point of view to estimate which is actually the density of the solvent in your solution. Okay, so this is why, in summary, this is why we have to stop share this one. This is why we have to set this parameter, which is the bulk range of the solution. What does this bulk range mean? It is the region in the solution which you believe from 10 on you don't have any more interaction, we don't have any more an effect of the presence of the protein in the structure of the solution, so it's outside the solute domain, outside the protein domain, and you stop considering the bulk domain at 15 angstroms, which means that this is an open subsystem which will be used to compute the density of the bulk solution. Okay, this is why this is going to be used. And if you don't put this option, the program runs, but it will warn you that it's a better idea to define manually this parameter. It is a little bit technical, but you face this rather early in the package usage, so it's better to explain it a little bit further. So, okay, here the computation ran. It took about 2 minutes, 1 minute 44 here, and we get an output like this one, which will tell you, okay, this is the concentration of the solvent in our simulation. As we can see, the concentration in the bulk solution is greater than the concentration in the complete simulation, which means that overall there is a depletion of the solvent in the vicinity of the protein. So the concentration in the bulk solution is greater than the concentration in the whole box. So there is somewhere else, which is in the protein domain, where there are less solvent molecules that one would expect. This implies the simulation concentration and so on, blah, blah, blah. And an important parameter, if you are computing the distribution functions, is to see if these distributions converged properly to 1. So we have the long range behavior of the distribution functions is expected to converge to 1. So we want to see things close to 1 here with fluctuations that are greater than the difference of these from 1. In this case, it has converged correctly. Okay, then we can save these results. This is explicit in the example, but here we don't need to do it. But you can save the results because this calculation can take a long time if you have a very long trajectory or a very large system. So we can save the results for further analysis. And now we are going to plot something. So the first essential plot that we want to compute. You remember that we have called the output of this calculation. We have assigned it to the results variable. So this results variable the first essential property that it contains is the results.d, which is the distances, and the results.MDDF. Okay, so here we have distance and MDDF, and this is This is glycerol, if I'm not mistaken. Okay, so this will produce the following plot. What we have here? Okay, the distribution function, the distance, this is for glycerol, and we see that there is, as I mentioned, a region where there is no glycerol, very, very close to the protein, there are no glycerol molecules because they cannot overlap with the atom of the protein. There is a significant accumulation of glycerol at short distances, and this is about the distance of a hydrogen bond, 1.9, 1.8. So glycerol is forming hydrogen bonds with the protein. And then there is a depletion region, and then the distribution goes to 1 at long distances. These small fluctuations are normal, and because this is a histogram, and the fluctuations depend on the width of the histogram. So, but this is not particularly important. If the fluctuations are around 1, it means that the distribution is properly converged. Of course, this is a sample simulation. If you perform the same calculation in a larger simulation, this thing will be anyway. Okay, this is the first result. So what can we do? So here we see, okay, glycerol is interacting with the protein, it's forming some interactions, and we can compute, for instance, which is the contribution, as I mentioned, that we can compute which is the contribution of specific atoms to this distribution. So, for example, we can compute the contribution of the polar residues. So, contributions, results, select, protein, polar. I don't know, it's not that. No, I don't remember how to do this. I have to look at the manual because I don't remember. That's been a while since I could run my own package. Atom and group contributions. You are not in my page. Let me share the complete. So it's easier to see where to find things in the menu. Here, there is. So okay. So I went to the manual, and I don't remember how to run the contribution functions. And there are a series of examples. So we have to provide this solid group or solvent group out, because we have to in order to decompose the contributions, we have to explain if we want contributions of solvent atoms or if you want contributions of solute atoms. So here we have, for example, solid group. Okay. So we want the contributions to be extracted from the results data structure of a solid group composed by these atoms. Okay, so we have defined this solid group object, and we have defined it with the polar atoms of the protein. And we obtained these contributions of polar atoms. And now we are going to plot in the same plot the distances and these polar contributions. This how is it called? Okay, this exclamation mark indicates that we are going to update the previous plot. So this is a Julia thing. And anytime, for any of the functions in Julia that have an exclamation mark, indicates that you are modifying some variable. So in this case, we are modifying the plot that is already available in the panel. So now we are going to plot the distances and the x-axis with the polar contributions, and then we can label polar residues. Okay, so this is the contribution of the polar residues to the distribution function. And of course, we see that they have an important contribution at the distances of the hydrogen bonds, which is sort of expected. We can compute the polar contributions or hydrophobic contributions of hydrophobic residues. The hydrophobic residues. Okay. This is the contribution of hydrophobic residues, and we can also plot again the contribution of the hydrophobic residues to the distribution function. Hydrophobic residues. You see, and of course they contribute much less because probably most of these hydrophobic residues are buried buried inside the protein, and they almost don't have any contribution at hydrogen bonding distance. There is some contribution at hydrogen bonding distance, probably because we have contribution of the backbone, and every residue can form interaction with the backbone. So we can add that plot the interaction of the backbone of the protein. So these are the contributions to the distribution function of the backbone of the protein. So we see here that, okay, the backbone forms hydrogen bonds with glycerol. There are backbone atoms that are exposed enough to the salt that form some hydrogen bonds. So, and we can go further, decomposing this into the contribution of different type of atoms, and we can also obtain the contributions of the, for instance, the oxygen atoms of glycerol. So we are going to the contributions in our results of the solvent group now, and of what? Of the glycerol atoms and element oxygen. So these are the contributions of oxygen atoms of glycerol to this distribution function. So which atoms are close to the protein? And you can add to our plot the contributions of the oxygen atoms. Glycerol. And you see, okay, the glycerol atoms of oxygen atoms of glycerol have an important contribution at hydrogen bonding distances, and you can go further to decompose these into very much detail as we want. Okay, good enough. There are prettier plots that we can do, which is we can one extension of this decomposition in groups is decomposing the contribution of every residue in the protein. So we could eventually run this command, computing contributions for every residue in the protein. However, we have already a convenience function for that, and you can compute the residue contribution of our results for our protein. You see, and this is plotted in first like this. So this is a function that will return an object which is a residue contribution object that's computed from within our results the independent contribution of every protein residue to the distribution function. And we got this Unicode plot just to illustrate a little bit what's going on. But we have a custom plotting function that will plot this as a pretty bidimensional plot. Which here there are too much residues there. We can set a step of 20 to see less indexes, to see less labels, and still okay. This is a little bit ugly, so we would want to understand what's going on around this residue 142. What can we do? We can rise this residue, the contribution, and plot the residues from 150 to, this is like slicing an array. you are slicing the residue contribution object, and then we are going to plot now only the contributions of the residues that are in this plot within the, in this case, 130 to 150, and now we got this image. Let's look at this small step. So now, okay, what's going on here? What's going on is that, okay, this is glycerol interacting with the protein, so we have here that it has a strong interaction with this tyrosine and with this arginine, and okay, that's pretty to see. But what if we wanted to understand the role of the backbone interactions here? We can do that. For instance, instead of, we can go on and get the residue contributions of the selection of the backbone atoms, which is the same thing, and then plot the same thing, but for the backbone atoms. So now we see that that tyrosine atom, which displayed an important interaction with the protein, is actually interacting through its backbone. And if you want to understand the contribution of the side chain, now we have the same thing, and this is the contribution of the side chains, and curiously, nice enough, we have the tyrosine now has a much smaller contribution. Let's remember, this is the backbone, this is the complete residue contribution, and now we have identified that the interactions of glycerol with this tyrosine are interactions through the backbone atoms, because this is the backbone contributions, and these are the contributions with the side chains. Okay. What can we do furthermore? Let's see what happens with water. So what's water? We have our selection of the water molecules in our system, and we can compute the results. Let's redefine what's the solvent in our system. Now our solvent will be the water molecules, which have 3 atoms each. And then our results, let's call them something else, are what? Results of water. And now this is the computation for the solvent is already redefined. The solute is the same as before. Now we are computing the interactions of the protein with water. And this will take about a little more than a minute. Let's wait. Why does this take more than computing a radial distribution function? Specifically because we have to compute that ideal distribution of atoms for every frame to be able to normalize the distribution functions, the minimum distance distribution functions. This is sort of technical. This is very detailedly explained in the papers. But we have, for each frame, we have to compute which is the distribution function, the distribution of the number of minimum distance found in the actual simulation, and this has to be normalized by an ideal distribution of the solvent, and we have to generate this ideal distribution of the solvent along this calculation, and this is why a minimum distance distribution function computation takes much more time than a radial distribution function computation. There is a much more sophisticated computation that needs to be performed to obtain the normalization of the distribution function. Very well. And then we can plot similar things. Okay, so everything went as expected. You see that distributions converged to very, very, very close to 1. So this is a good sign. Let's plot again. This was the distribution function for glycerol. Let's plot now the distribution function for water. Now that we have completed now, sorry, I want to see that. So we see that, okay, water has a very, very sharp peak at hydrogen bonding distances, and then it is, so the fact that in this region water is has a density smaller than the density of the bulk is probably associated with the fact that the glycerol is occupying all this region by interacting with the surface of the protein, thus excluding out water and other glycerol molecules when it is forming a hydrogen bond. So glycerol form a hydrogen bond here. We'll see this later in the density plot, but glycerol is forming hydrogen bonds with water, and that excludes other molecules from the region where the rest of the glycerol molecules is occupying nearby the protein surface. Again, we can compute, anyway, anyone? Okay. Again, we can compute contributions. So let's see, the backbone, let's compute the contributions for, in case of the backbone, but for the water. So the backbone, this is water backbone. So there it is. Okay, of course, water can form hydrogen bonds with the backbone, and there it is. Forms hydrogen bonds with the backbone. Very nice. And we could go on decomposing these into the atoms of water, the atoms of the protein, and the groups of the protein, and so on. But let's go directly to the residue contributions of water, which will be the contributions, the residue contributions of our water results of the protein. And we have the same sort of plot. We can be plotted by this command. And again, we have the same sort of profile. We can now plot the range between 100 with the step 2. We see the water interactions with the same region of the protein. Now, curiously, we see that glycerol had a strong interaction with this tyrosine residue here, and water doesn't have strong interaction there with the tyrosine residue. So they are different in that sense. And what can we do actually? We can actually confirm that, because we can do the difference between these two residue contributions. So we are going to compute the difference between the residue contributions of water to the distribution function minus the density of what I have called this. I think I have called just like that. So this is what generates this plot, and this one generates the same plot but for the contributions of glycerol, which I haven't subscribed by anything. So, and this is the difference in the density of water minus the density of glycerol in this region of the protein. And we can plot with the same command the same plot. And we see that, for instance, where is red points indicate that the density of water is greater than the density of glycerol, and the blue points indicate that the glycerol is more concentrated than water. And we see that in the region around that tyrosine, this is mainly blue, which means that glycerol has a stronger affinity for this region than water. Okay? But remember here that we have much more water than glycerol in this system. So the regions where glycerol is blue, it means that there is a very large accumulation of glycerol there in order for the density of glycerol molecules to be greater than the density of water, even considering the fact that the concentration of water in the system as a whole is much greater than the concentration of glycerol. except that here we are computing the difference between distribution functions which are already normalized by the data. So this is I use that to mention that we can compute the coordination number. So to correct what I have already said, so this is a relative affinity. So the region where we have blue dots imply that the affinity of glycerol is greater than that of water in that region. And if we want to compute something that's less thermodynamic but more simple to understand structurally, we can compute the coordination number around each residue, which in this case can be computed by a similar command. So I will call it coordination number of water around the protein, and here I will provide this flag, which will then compute the coordination number. I will again plot again. You see this plot is quite different. So what's happening here? Now we are computing the coordination number. Let's show it for the range that we are plotting. So what's going on? This is the colors indicate the number of water molecules that are within each distance of each residue of the protein. And this of course increases, because as you go farther away from the protein, the volume of the solution increases as the radial distribution function. Or if there is a spherical, or in this case not spherical, but there is a volume that increases as we go further away from the protein, which implies that the number of water molecules that are being counted increases. The distribution function normalizes that by the distribution, but here we have the coordination number, which is simply the count of the number of water molecules that are present at this distance. So again, okay, it increases and there is an accumulation around some residues. We can now, this is the coordination number for glycerol. Let's compute the same thing. We will have called these results. And again, we can compute this same plot, but for glycerol. And we see, okay, there is an accumulation of tyrosine molecules in this region, which reflects that peak in the distribution function that we observed earlier. Now, the differences in coordination numbers is something that is easier to understand and will help understand the differences in. So now I compute the difference in the number of water molecules that are within each distance from these residues minus the number of glycerol molecules that are within each distance from these residues. And this plot is almost completely red. Why it is almost completely red? Because there are much more water molecules in the system than glycerol molecules. There are much more water molecules in the system than glycerol molecules. So the number of water molecules is almost always greater than the number of glycerol molecules around each residue, except, for instance, around these residues, and these residues are probably buried inside the protein, so there is no approximation of either glycerol or water. And now there is here something that's associated to the scale only, because we see in this first approximate map that there is a blue dot here. This blue dot here could appear here around tyrosine 143, which is the residue that has this greater affinity for water. And we can adjust that, adjusting the limits of this plot, which I have not tested, but I will try some values here and try to plot this using limits that could capture that difference there. Too much. Okay, this is getting prettier. This is getting even prettier. Okay. So now we have the same density map, but in a different color scale. The accumulation of water has blow up the color scale. So here we have a strong red region indicating there is much more water in this region than glycerol. But in these colors, with this color scale, we can see that around tyrosine 143, in fact, there is a greater number of glycerol molecules than water molecules. And this is associated with the much greater affinity of glycerol to this tyrosine residue that we could see in the other plot. This is not right. Oh no, this is why. This is only the side chain. This is why we're not seeing. This is only the side chain. So now we're going to see this clear. I have, okay. So we can see that there is a much greater affinity of glycerol around this tyrosine residue, which is reflected even in the fact that the number, which is computed by the coordination numbers, is reflected in the fact that even if we have much more water in the system than glycerol, still the number of glycerol molecules around this residue is greater than the number of water molecules in the same region. Okay? Okay, so this is an overview of the first example. Actually, much more than, okay, this is the first example. So protein, water, and glycerol. We have shown how to compute these maps, and we can also compute three-dimensional maps with the same information. And there are some detailed instructions here, but maybe I would prefer not because there is a video explaining how to produce a three-dimensional map. The video is quite bad, but anyway, it's what I produced so far. So, but yeah, I think you can follow them. The details are here. And then you can produce images of, like this one, in which we'll see the density of atoms around the protein in a three-dimensional way.